After incubation, there is obviously microbial growth on the Petri plate, including a number of individual colonies that we can assume to be composed of a single species. In order to get a DNA sequence from the putative pathogen, we'll first need to extract DNA from it. There are many ways to do this, but for today we'll be using PrepMan Ultra. We've already put 50 microliters of PrepMan Ultra in the tube, and we'll simply transfer bacteria from a single colony on the plate into the 50 microliters of PrepMan Ultra. The tube is then incubated at 100 degrees Celsius for 10 minutes, after which it's ready to be used as a DNA template for PCR. PCR, or the polymerase chain reaction, is able to amplify specific targeted regions of DNA to allow for their subsequent sequencing. We'll be running 3 reactions today. One, a negative control, which we do not add DNA to. This will help us detect any contaminating DNA in our PCR reagents. Second, a positive control, which we add DNA from a known source to make sure that our PCR works. In this case, we've added DNA from a known soft rot bacterium. For our third reaction, we'll use the sample of DNA that we extracted from our unknown microbe. In order to conduct PCR, each of the 3 reactions will need a number of reagents, as detailed in the recipe in the right column. However, instead of making each reaction individually, we'll instead make one master mix with 3 times the volume for a single reaction, then distribute that volume between the 3 tubes equally. Add the appropriate reagents one at a time to the master mix, as detailed in the PCR recipe. Then, distribute an appropriate volume of master mix, in this case 49 microliters, to each of the 3 reactions. Finally, the DNA sample extracted from the unknown bacterium is added to the sample reaction. The sample from the known soft rot bacterium is added to the positive control reaction, and water is added in place of DNA template for the negative control reaction. All 3 samples are then placed in a thermocycler. A thermocycler is a programmable machine that can cycle the reactions through a number of precise temperatures for precise durations, which allows the reactions to pass through the cycled temperatures for denaturation, annealing, and extension needed for successful PCR.